hydroxyl radical, and then other ones. What I'm going to talk about today is mostly superoxide. You'll hear me refer to hydrogen peroxide and hydroxyl a little bit later on in the talk, but we're really going to focus primarily on superoxide. So ROS kind of have a negative connotation, a kind of a bad reputation, if you will. So probably the first thing that came to mind when I mentioned ROS is stress and maybe antioxidants. And so they kind of have this bad reputation. And so, and this is best shown in this New York Times article that I found from 1993. I love this article, "The Price We Pay for Breathing." And I'm just going to read off this one little clip from this that says, "In the age of modern triumphant, all things radical bear a bad connotation, but surely nothing can match the ill repute of free radicals." And so I just love that little clip from this article. And you know, this really rings true when you think about reactive oxygen species. And you know, and there's some reason for that, and that's because ROS have been linked to a number of different diseases within a number of different systems, including plants and animals, including the human body. And so various forms of cancer, diabetes, premature aging have all been linked to elevated concentrations of reactive oxygen species within the body. But what you may not know is that reactive oxygen species are also essential for your body and other organismal systems. And so if you go on and read a little bit farther down in this article, you'll see it says, "Yet, like so many of society's maligned, free radicals are often misunderstood." So this is kind of what I want to talk a little bit about today, is this idea that, you know, we don't have a really good understanding of that beneficial process of reactive oxygen species within marine systems and how it's important for marine organisms in general. So to kind of start to springboard into that, I wanted to first talk a little bit about where reactive oxygen species form. Well, first we'll talk about a bacterial cell. So this is my really poor depiction of a bacterial cell, just to try to show that there are two, you know, compartments that we're going to talk about. So you have to think about when we're talking about intracellular, there's an intracellular compartment and an extracellular compartment. So when we're thinking about these negative consequences of reactive oxygen species, primarily what we're thinking about is this intracellular compartment. And so that's inside the cell. This is linked primarily to respiration and photosynthesis. And this is when concentrations get really high, it's elevated due to stress. And so what I'm showing inside is you see superoxide, hydrogen peroxide, and hydroxyl radical that can form, and they can transform from one to the other inside the cell. And so that's that intracellular compartment. There's also the extracellular compartment outside the cell where ROS are formed. And this is believed to be more essential, intentional formation, and involved in a diverse set of physiological processes. And what I have showing there is hydrogen peroxide and the arrow moving in and out of the cell. So out of all of the reactive oxygen species that can form, hydrogen peroxide is the only one that can pass biological bilayers or membranes. And so the other ROS can't pass through the biological membrane. So if superoxide forms inside the cell, it can't pass outside the cell. So if superoxide's found outside the cell, it had to be formed outside the cell. And so, so if superoxide is formed inside the cell and it gets to elevated concentrations, it has to be also dealt with inside the cell. And so cells have an ability to highly regulate the concentration both inside the cell and outside the cell. Now, as I mentioned, extracellular superoxide and ROS production outside the cell is believed to have this diverse physiological role within different organismal systems. And in fact, it's been shown that there's a suite of enzymes called transmembrane NADPH oxidases, or NOx enzymes, that have been shown in eukaryotic organisms that are responsible for extracellular superoxide production. And so these have been found in fungi, in plant and mammalian cells. And so this extracellular superoxide production is formed when NADPH inside the cell is an electron is extracted by these NOx enzymes and transferred to oxygen outside the cell. Now these NOx there's several of these different NOx enzymes, different isoforms of these enzymes, as well as other forms called duox that are throughout the different eukaryotes. And it's believed that this extracellular superoxide is beneficial for the organism and involved in a number of different physiological processes like cell signaling, cell differentiation and growth, cell defense, the immune response, and wound repair, just to name a few. And so, as I mentioned, this has been shown in a number of different systems. This has been shown for primarily in fungi and plants and mammals. And so what we really have in these different systems, in these organismal systems, is what we like to think of as the Goldilocks scenario, where if concentrations of superoxide, both inside and outside the cell, is too low, then cells can't actually do the essential physiological functions that are required to grow. Can't differentiate your cells, you can't fight off pathogens, you can't induce wound repair. But if the concentrations get too high, then you induce oxidative stress. So this is where you get lipid peroxidation, oxidative damage to proteins and membranes. And so really cells have to work to keep that concentration of superoxide right in that concentration that's not going to damage cells, but enough that you can have the right concentration to have these physiological functions met. And so we have, again, this Goldilocks scenario of having just the right concentration. But when we think about marine life and what we know about in marine systems, we really haven't gotten there to be thinking about this in this Goldilocks type of scenario. Instead, we focus primarily on this negative aspect. When we think about superoxide in the ocean, these are the types of terms that come to mind: oxidative stress, DNA, protein damage, lipid peroxidation, and cell death. So although that we know a lot about the essential role of reactive oxygen species in organismal systems, within terrestrial systems, within the medical literature, that just really hasn't transferred over into the marine realm. Now, I should say that there's been a lot of work that's been done in pathogenic strains, such as Chatonella has been studied for many years, studying extracellular superoxide production. But in your typical marine phytoplankton and marine organisms, this just hasn't been something that has been really well studied. That is until the early 2000s, where a couple of groups showed that two of the common phytoplankton, Thalassocyra and Synechococcus, produced extracellular superoxide outside their cells. So these are two of our common phytoplankton in the ocean, a diatom and a cyanobacteria, that were producing superoxide under optimal growth conditions within the lab and producing it outside the cell. And these are at concentrations, as you can see at the bottom of the picture, that are at rates that were both physiologically and environmentally relevant. And I know an attomole per cell per hour might seem like a slow rate, but if you're thinking about that on a per cell basis, this is actually a rate that's actually relevant for that organism and for environmental fluxes within the environment. And so this was really kind of the beginning of thinking about, okay, well, this is a process that we don't really know a lot about in the ocean. This is a process that must be playing some sort of role within organismal physiology, within marine phytoplankton, but we just don't know anything about it within marine systems. This is about the time when I started getting interested in reactive oxygen species. And so I wanted to try to understand, is this a broader process within marine phytoplankton, within other organisms, marine microorganisms within the ocean? And so I started springboarding off of the work that was done by Adam Kuska and Andrew Rose here with these two first model organisms. And so before I dive into that data, I wanted to first kind of give you an indication of how we make these measurements. So the way we measure superoxide is that it reacts with, well, the primary way that we measure it is that we measure it by reacting it with a chemiluminescent probe. So this is a chemical that when it reacts with superoxide, it gives off chemical light. So the trick with measuring superoxide is it has a really short lifetime. It has a lifetime in the ocean, typically of less than a minute, most of the time about 30 seconds. In cells and within the body, it's much less than that. But we're looking at the extracellular concentration. So, we're dealing with typically about 30 seconds that we have to deal with. So what we want to do is we want to use a flow injection system where we're flowing water past the cells, and then we inject it into this instrument where it has a mixing cell, and that mixing cell takes in the effluent that went past the cells, and then it reacts it with this chemiluminescent probe. When it reacts with that probe, it gives off chemical light. And then we measure that chemical light using a photomultiplier tube. And then we watch that signal over time. And then we can convert that chemical light into concentration by making superoxide in the lab, either chemically or photochemically, and then relating that signal to concentration. So I'll give you just an example of what one of those traces would look like. So this is chemiluminescence on the y-axis versus time on the x-axis. And you're looking at a filtered seawater that was a sterile filtered seawater that was flowing past the cells. And they're typically in some sort of little Swinex holder or some sort of little flow cell holder where the cells are added. So we typically have the filter holder in there to begin with, then we flow the filtered seawater through, then we stop the pumps, add cells, restart the pumps, and we get the steady state signal. And then we stop the pumps again, and then we get another steady state signal, then we'll stop the pumps, add cells again, get our third steady state signal. And then when we're happy with our experiments and we've run all the number of cells that we want, we add a scavenger called superoxide dismutase, which is an enzyme that specifically reacts with superoxide. And then we'll see our signal go away, and then we know that we've been measuring superoxide. So at the end, what we have are a number of different signals. At the very bottom, you see we have this probe auto-oxidation that we have to remove from all of our signals. And then we have the first steady state superoxide signal, and then the second, and then the third. And then, as I said, we can turn that signal into superoxide concentration by having our calibration standard that we've made either chemically or photochemically. So over the years, over the last 15 or so years, we've been kind of picking away at this in the lab by studying organisms we either had in my lab. I've also been calling up friends and colleagues, asking them to send me their favorite pet strain. And we've been measuring pretty much every organism we can get our hands on to try to get just some broad representation of how widespread is this ability to produce extracellular superoxide within marine organisms. And so what you're looking at is a compilation of that data from over the years. And what this represents is a large taxonomic and ecological diversity of different bacteria and phytoplankton. On the y-axis is the superoxide production rate in attomole per cell per hour. So this is normalized on a per cell basis. And you need to note that the axis is a log scale. So you're looking at a pretty big distribution of rates. And then these are all representing different species. And I know you can't see who they are, but I can tell you that they represent a lot of a range of different species. And this includes a lot of the common organisms that you think about as major players in the ocean, including Prochlorococcus and Synechococcus. This includes Thalassiocyra, SAR11, a number of different roseobacters, and so on. All of these organisms were grown in the lab using what was thought to be their optimal growth media, grown to a mid-exponential phase. And I need to point out that these are light independent rates. So even for the phytoplankton, we grew them up in the light to their optimal growth phase. And then we did the superoxide production rate in the dark, because what we want to do is compare dark production rates among all the organisms. The phytoplankton always make more superoxide in the presence of light, which I'm not going to focus on the phytoplankton today, but so what we're only going to be talking about is light independent superoxide production. So this is a dark rate. So what we have are these dark production rates as a function of the different organisms. One of the interesting things that kind of popped out over the many years, and this was widespread regardless of the type of organism, was that we saw this inverse relationship between extracellular superoxide production and cell density. And so here you're looking at Synechococcus, Phaeocystis, and Thalassiocyra, so different plots of those organisms grown. And you're looking at superoxide production rate as a function of cell number. And what you can see for each of these organisms is that the superoxide production rate per cell was higher when they were at lower cell numbers than when they were at higher cell numbers. So on a per cell basis, so that indicates to us that they were producing at a faster rate when they're at lower cell densities. And when we first saw this, we were really excited because what this was reminiscent of was a cell signaling type behavior, so like a quorum sensing type behavior. And we looked at this more with focusing in on two different bacteria within the Rosiobacter clade. So this is Rugeria pomeroy, DSS3, and a Rosiobacter that I isolated during my postdoc, Rosiobacter AZWK3B. And these two organisms show the same behavior when they're at low cell densities. They had a higher cell-specific superoxide production rate than at higher cell densities. So when we're looking at these plots, though, the way that this was done was, as I showed you with that that luminescent trace, where we're sequentially adding more and more cells into that flow cell. So what I wanted to do is said, well, maybe this is an artifact where we're just adding more and more cells. Maybe we're crowding that flow cell. So what I wanted to do is take a step back and say, okay, well, what we should do now is grow them up to mid-exponential phase and now dilute them instead. So dilute them out in sterile seawater and see when cells get diluted, do they upregulate superoxide production when they're in lower cell densities. So we did that with these two organisms. And so what you're looking at on the left is the steady state superoxide concentration as a function of dilution. So on the left, for those two organisms, you can see that when they're at low dilutions, as you would expect, you'd have higher superoxide concentrations. So they're not diluted, so the superoxide concentrations build up. When you dilute it out, there are lower concentrations of superoxide. However, when you look at that as a superoxide production rate on a per cell basis, on the right-hand graph, you can see that actually at higher dilutions, each cell was actually producing a higher rate of superoxide than in the lower dilutions. So this actually happened really fast. So we did this as a function, diluting them out and measuring after 3 hours and then measuring again after 7 hours. And the organisms were actually responding within 3 hours and producing at a faster rate within 3 hours, and then producing even more at a faster rate again at 5 hours. So this indicated to us that they were responding to these lower cell densities and increasing their rate of production. So we are finding this all very intriguing. This is indicating to us that there's some regulation on this superoxide production outside the cell. So we wanted to explore Rugeria DSS3 in a little more detail. So we decided to study superoxide production along a growth phase as a function of growth phase. So what you're looking at here is a plot of Rugeria superoxide concentration as a function of growth phase, which are the blue dots. So you're looking going from lag phase through exponential growth up through stationary phase. And what you can see is superoxide concentrations continually build up within the flask until Rugeria hits stationary phase, and then superoxide concentrations are gone. So this was actually extremely exciting and kind of rewarding to us, because over the years, one of the kind of the criticisms we've gotten was that, well, maybe the superoxide production or the superoxide that you're seeing is just lysed cells, right? That this is just all intracellular production, the cells are lysing, and you're seeing superoxide because of that. And if that was the case, then we would see a lot more superoxide in stationary phase than we would during lag or exponential phase. So that was a nice result to find. So the one thing that we wanted to follow this up with was, okay, so the steady state superoxide concentration outside the cell is going to be a function of both production and decay. So we don't know whether or not the organism is regulating its production rate or regulating its decay rate, or both. So we wanted to see whether or not decay was actually being regulated as well. So the next thing we did was took the organism at different time points. We spiked in a known concentration of superoxide, and then we looked at the decay of that superoxide over time, and using a decay function, we calculated out the kinetics of that and came up with a decay rate constant. So what you're looking at now is the decay rate constant through the growth phase. And so what you can see during lag and exponential phase, the decay rate constant is really low, indicating that the Rugeria was not decaying the superoxide, wasn't expressing, or the SOD was not active. Yet once Rugeria went into stationary phase, the superoxide decay rate went through the roof. And actually those little bars, or those little hatch marks on top of the bars, are there to remind me to tell you that those aren't the actual end of those values, because the decay was so fast within those last two time points that I couldn't actually add enough superoxide to actually get a decay rate. It was decaying so fast that I couldn't actually measure it. So apparently, and obviously, the decay rate was so much faster than my ability to measure it. And so superoxide or Rugeria apparently did not want superoxide around. So when we think about this in a different way, the concentration, we can now look at the concentration on a per cell basis. So this is now manifested as a function of both the production rate and the decay rate now. So what is the concentration per cell? And it's striking that the concentration on a per cell basis is surprisingly constant all the way through the growth cycle, all the way until Rugeria enters stationary phase. So the organism, through controlling both the production rate and the decay rate, is keeping that superoxide concentration incredibly stable until it enters stationary phase. And then the concentration just plummets and decay rates go extremely high. So that's telling us that it wants to have a really tight regulation on superoxide, and it wants superoxide outside the cell during growth. So what we wanted to do is say, okay, well, during growth, can we remove superoxide then by adding extracellular, by adding exogenous superoxide dismutase, and see how that would impact growth? So is this going to have an impact on growth of Rosiobacter? And so we added an exogenous superoxide dismutase. And so we did this for both Rugeria and Rosiobacter. And so what you're looking at are their growth curves in the absence of superoxide dismutase, so just their normal growth curves. And then what we did was we added different concentrations of superoxide dismutase, and then looked at how that impacted their growth curves. And what you can see is by adding superoxide dismutase, we got an increasing inhibition of growth for these organisms. So I can't tell you what exactly superoxide is doing, unfortunately, at this point. So this is something we're actively working on still, trying to figure out what superoxide is doing outside the cell. But what it appears to be happening is that superoxide is somehow playing a role in growth regulation in these two Rosiobacter species. Interestingly, this has also been shown in a pathogenic strain of E. coli. These same results have been shown where if you scrub superoxide outside the cell, E. coli can't grow. And like everything, everything's known about E. coli. So the enzyme has been worked out for what the enzyme is for E. coli that makes it extracellular superoxide. And they've shown that if you upregulate extracellular superoxide production, E. coli, the organism will just grow like mad to extremely high cell densities. If you knock that gene out and it can't produce extracellular superoxide, then E. coli can't divide. So there's been a number of different suggestions on what superoxide could be doing. It could be reacting with the outer membrane. Superoxide's extremely reactive. It could be reacting with the lipids and impacting the biophysics of the outer lipid bilayer. It could be reacting with some outer proteins, changing their conformation. It could also be dismutating, forming hydrogen peroxide, which can then diffuse back inside the cell and act as a signal. That's been shown to occur in fungi. So right now we don't really know, and something that we're actively exploring, but kind of interesting and a fun thing to be looking into, knowing that now there's some sort of link between superoxide and growth in these two organisms. We have been starting to figure out what enzyme is involved. So in both Rosiobacter as well as other organisms. And I'm not going to dive into a lot of this today because there's not enough time, but we're finding that while NOx is known to be involved in extracellular superoxide in eukaryotes, within Rosiobacter, we have found that within at least one organism, there's a heme peroxidase that's involved. And we've been doing this using native gels and using specific stains that will bind when it reacts with superoxide and extracting those and sequencing those bands. And then thalassiocyra, we've recently shown that it's a glutathione reductase. And these are soluble secreted enzymes that are outside, that are anchored on the outer membrane. But we're just beginning to scratch the surface on this. And so all we can say right now is it looks like there's a diversity of extracellular enzymes that are being used by organisms and that are soluble enzymes, including also these transmembrane enzymes like the NOx enzymes that are involved. So it seems to be much more complex than what we had thought and we're hoping was going to be involved. But to continue on, I wanted to talk about, well, we know that extracellular ROS is now regulated by microbes within marine organisms. We believe now as part of their central physiology, it's playing some sort of role in growth, at least in some organisms. So what is the role in the marine ROS concentrations and in the flux of ROS within the ocean? And so what do we know about ROS in the ocean? So if we think about reactive oxygen species in the ocean, the canonical view was that the primary sources of reactive oxygen was from the photoexcitation of color dissolved organic matter. And so light reacts with color dissolved organic matter and excites it, and that reacts with oxygen, produces ROS. And this has been known to be a primary pathway for ROS for many, many decades. But that really constrained ROS to kind of those surface sunlit waters and also constrained areas to really organic rich environments. And so it was really believed that ROS were really going to not play an important role kind of in more oligotrophic regions as well as, kind of deeper within the water column. But now we know that, our data from our group and other groups are now point that to, the importance of microbes for extracellular ROS and that they could be an important source of superoxide as well as other ROS to the ocean. But what do we know so far and how far have we come in understanding them as sources to ROS within the ocean? As I mentioned, superoxide has a lifetime of less than a minute. So this has made it incredibly challenging to be able to measure these ROS within various systems. So, think as oceanographers, what we do is we take a rosette, we throw it overboard, we collect water in Niskin bottles, we collect those bottles, we hook it up to our instrument, and then we measure, those analytes. But by that time, superoxide's turned over many, many times. And at as of just until recently, we had no in situ instruments to be able to measure a reactive oxygen species in situ. So the approaches that we've used over the last couple decades were that we would collect water, we would take that water and we would incubate it in conditions that we hoped mimic the in situ conditions as close as possible, the temperature conditions, the light conditions, and then we would be able to then at least measure those steady state values for those incubations. The other approach was we would take those waters and then we would filter them and we would measure the decay kinetics of superoxide or hydrogen peroxide. And then we would get a particle associated decay rate, which then we could back calculate out to a particle associated production rate. And so these are the kind of the two primary ways that we've collected superoxide and concentrations and rates over the years. And even with that, we only have a handful of measurements within the ocean. With that being said, there has been quite a few studies that have pointed to the importance of particle-associated superoxide production, both within the surface, ocean, and in aphotic waters. And these, again, are light-independent particle-associated. So this is that dark source. And so these were, again, where waters were taken, they were filtered, and in the dark looked at the decay kinetics and then back calculated out to get that decay kinetic, what would the in situ production rate had to have been. And so here's some data from a study done by Andrew Rose in the Costa Rica Dome. And what you're looking at is a water profile, and the black circles are superoxide production rates in the dark for particle associated production rates. And what you can see is that there's a kind of a peak where the chlorophyll max is, but that there's still superoxide production rates well below the chlorophyll max and well into the dark and into the aphotic zone. And similar studies have shown this in other areas, including the Gulf of Alaska and other regions throughout the ocean. We've followed up on this over the years in trying to not only show that we have this light independent superoxide production, but how much of this superoxide production is particle associated versus not particle associated. So this is some data from a recent cruise that we had on the eastern seaboard, which is shown in the map on the upper right. A number of different stations along the coast, and then one off-shelf station. And this was during coastal bloom, and so we had pretty high productivity. And so you're looking at coastal Maryland, two of those stations, coastal Maryland and coastal New Jersey. And the black bars are the steady state superoxide concentrations. And so these are dark or light independent superoxide concentrations. Now, if we look at how much of that is particle associated versus not particle associated, the dark blue bars are how much is particle associated. The light blue bars are not particle associated. And you can see that not only does it vary from site to site, but it varies within site. So for coastal Maryland, most of it was particle associated. All the bars are blue all the way down. And I should point out that the top in the white is the photic zone, and the gray on the bottom is to indicate where we're less than 1% of par. So that's kind of the definition of where you've entered the aphotic. So that these are considered dark waters. So what you can see is that we have superoxide all the way through the water column. Oh, I don't know why the depth numbers aren't on there, but this was going down to about, I think this was about, I'll have to look up. I think that was going down to about 60 meters. And then on the right for coastal New Jersey, we have, again, superoxide being produced all the way in the photic and the aphotic zone. But here, you see this not particle associated superoxide playing a bigger role, and it changes from depth along, depending on the depth and within the New Jersey site. For the off-shelf station site, which is indicated up here by this circle around this dot here, we have superoxide being measured all the way down to 2,000 meters, so very deep into the water column. And interestingly here, we have superoxide being produced primarily by sources that are not particle associated. So what would these be? These are things like dissolved metals. These would be reactive dissolved organic carbon. We could maybe have sources such as extracellular enzymes that are playing a role. But again, we just really don't have a good handle on what these sources could be. And so this is an area of active study in a number of different groups, trying to understand what are these sources of superoxide and other reactive oxygen species within these systems. So, what we're starting to find is that, superoxide occurs throughout the water column, but it has complex distributions and a diversity of sources that we just really don't have a handle on yet. But one thing we do know is that, particles appear to play a dominant role in some ecosystems, but we don't really know what those particles are. Some of that is going to be phytoplankton, some of that bacteria, minerals could also be playing a role, but we don't know exactly which organisms and how they're doing it. And so we're really constrained right now by our ability to have to do these incubations in the lab. We're constrained by our ability to have to do these particle associated rates rather than having true in situ concentrations or true in situ rates. Furthermore, we know that some ROS is being formed by point sources, not necessarily just microorganisms within the waters. There's going to be point sources of superoxide and other ROS that we're going to want to measure in the water column and not necessarily just within the water. One of those being corals. So corals have been shown to be able to produce hydrogen peroxide. We've recently showed that they can also produce superoxide. And in fact, reactive oxygen species have been accused of being one of the primary reasons why corals are currently bleaching, and ROS are accused of breaking down the essential symbiosis between the algae and the coral. And so there's a great need to understand how reactive oxygen species are being produced within coral reefs and coral reef ecosystems. And yet we really don't have any good measurements of corals, of reactive oxygen species within corals in situ. So over the last 4 or 5 years, my group has been working on developing new sensing technologies for measuring ROS at various scales. So all the way from the ecosystem scale down to the cellular scale. So from the deep sea to shallow coastal to cellular. And so today what I want to, I'll just end by talking about these top two, the deep sea and the shallow coastal sensors that we've been developing. And the first one is the DISCO. So DISCO is a diver operated submersible chemiluminescent sensor. And it's a handheld sensor that's rated to 30 meters. And it's based off on a lot of the technology that we use for the lab-based instrument, where it's a flow injection system. It works on the chemiluminescent probe, and it has a tablet that's oil compensated, so we can control the DISCO with our finger, and we can have a live interface to see when we have a signal with the coral. The DISCO got to have its debut a few years ago at the Gardens of the Queen in Cuba, and made its first measurements within a number of different reef sites along the Cuban coast. And so this is an example of what one of those traces would look like. So what you're looking at is the raw signal, or the lumens, as a function of time. And the scuba diver will jump in the water with DISCO and measure background seawater, which you see on the far left, and then swim up to a coral surface and take the DISCO's wand and hold it up to the coral surface. In this case, it was Orvicella faviolata, and didn't see a signal. Go back to the background seawater, and then go back to the coral surface and didn't see any signal. Go back to background seawater. went to another coral, this time Porites, and then we saw this big signal. So this big spike in the concentration or the signal of superoxide, and then go to background seawater and so on, and then go back to other corals, and then continually go to the background seawater. And then occasionally we would pump in SOD to confirm that we were in fact measuring superoxide. In this case, you can see that the background seawater was kind of inching up. Our original versions of DISCO was having some heating issues with the electronics. So our baseline value kept creeping up as we were going along in the dive. So we would always make our measurements for the corals as a function of the initial and baseline of the seawater. So we have all of our concentrations normalized to this seawater baseline. The new version of DISCO doesn't have this issue. But if we take all this data and then we compile it and we have these seawater normalized superoxide concentrations, what we would find is that we had this species-specific superoxide levels associated with corals. And so for some corals, like the Porites species, we had these enormously high concentrations of superoxide at the surface, like up to 200 nanomolar levels. And these are levels that are way over what we typically see in the ocean. And we never see anything above, you know, a couple nanomolar in the ocean. So we were getting up to hundreds of nanomolar. And then for some species, we were seeing 0 or below background seawater. And so we're really excited about this data, and I don't have time to jump into all this today, but you know, we have some ideas on why this is happening, and we're starting to explore coral larvae and what are kind of some of the underlying sources and mechanisms responsible for some of these differences. But to move in to the other instrument, we have Solaris. Solaris is the DISCO, but kind of taken to the next level. Solaris is rated to 4,000 meters. It can integrate into both a CTD and onto vehicle platforms. And because Solaris can go all the way down to 4,000 meters, it has to be able to also be able to do in situ calibrations. So it has the ability to make superoxide in situ using an LED photochemical platform. And so we got to the chance to be able to use Solaris for the first time last year on an Atlantis cruise, where we got to dive at the Davidson Seamount at the Monterey Bay National Marine Sanctuary. And that's the wand of Solaris, taking a measurement off of Paragorgia, which is a deep sea coral. And here again, what you can see is the seawater signal, and then up near the one arm of the coral, and then over to another segment of the coral, and then back to seawater. You can see that we have this large spike in the signal. And similar to what we saw with the shallow corals, we have this large species variability in deep sea corals. But interestingly, you know, deep sea corals are also making superoxide at, you know, an order of magnitude lower level than what we were seeing with the shallow corals. But that's to be expected, since the temperatures are much lower and their metabolisms are much lower and growth rates. So interestingly, you know, the algae are what are thought to be responsible for a lot of the intracellular superoxide dynamics in corals, and thought to be responsible for causing the bleaching of corals and this intracellular superoxide process. However, deep sea corals don't have any algae, and so we have extracellular superoxide being formed, you know, kind of pointing to the coral animal as the source of the superoxide, as well as potentially, you know, the bacterial symbionts. And so we're currently exploring this a little bit more. However, coral also have NOx, so they have the potential to make extracellular superoxide. So, you know, I'm kind of starting to wrap up here. I know I'm running out of time. What I wanted to point out is, you know, what we have are seas of superoxide. So I'm hoping that you're kind of getting the sense that, you know, this is not just, you know, just a one-off of organisms. We have lots of different sources of superoxide I didn't talk about today, but seaweeds are also sources of hydrogen peroxide and superoxide. We have abiotic sources like reduced metals and hydrogen sulfide, reduced carbon and corals. And there's just a lot of reactive oxygen species within the ocean. I tried to point out that this is important for the physiology of organisms, but in thinking beyond that, this is important for just biogeochemical cycling in general. Reactive oxygen species are important reactants. They can serve as both reductants and oxidants. So superoxide can actually be both a reductant and oxidant depending on what it's reacting with. Hydrogen peroxide in the center here, if it reacts with even small concentrations of iron, it forms hydroxyl radical. And hydroxyl radical can oxidize pretty much any carbon it comes into contact with, including recalcitrant forms like lignin and cellulose. So it's believed to be a primary pathway for remineralization within many ecosystems. Superoxide can reduce iron-3, forming iron-2. And it's actually been suggested that maybe that's why phytoplankton are producing superoxide. Maybe that's a primary reason, is that that's a mechanism by which to make iron more bioavailable. I got into this whole mess of studying reactive oxygen species because I was studying manganese oxidation. So superoxide actually oxidizes manganese-2 to manganese oxides. Manganese oxides are one of the strongest oxidants in the environment. And so I'll just show here before I wrap up that manganese oxides have been shown to be formed by a number of different organisms via this pathway, including Rosiobacter, which is a marine bacterium that I talked about earlier, and then soil bacteria, algae, diatoms, most recently. A number of different fungi have been shown to form manganese oxides and also manganese-3 via the formation of superoxide. And so this extracellular formation of superoxide can induce this cascading impact on many different metals. It's been shown to be a primary way that copper gets cycled in the environment. And then hydrogen peroxide similarly will react with a number of different oxidants or a number of different metals. So there's a whole area of research to go into, kind of this, this domino effect of how these reactive oxygen species are impacting other biogeochemical cycles. If you look on the flip side too, on the front half of that reaction, we just recently got very excited thinking about what is the role of just the formation of reactive oxygen on the oxygen cycle itself. And so we have a paper that just came out looking at what happens to the fate of superoxide. So on the bottom here, if you think about superoxide gets formed from oxygen. So oxygen's getting consumed. Now, where that superoxide goes will determine whether or not oxygen gets consumed completely, so gets lost, or whether or not oxygen gets reformed. And so if you go to hydrogen, oxygen or superoxide can either go to hydrogen peroxide to water, where you'll get complete loss of superoxide, or superoxide can ultimately come back to oxygen. By looking at our light independent superoxide production rates that we had from a suite of different organisms, and then by using the distribution of kind of the key organisms in the ocean and the maps of those distributions of those organisms, we then calculated out how much this could impact the global cycle of oxygen. And we calculated out that this could represent a gross oxygen sink of up to 35% of the marine gross oxygen production. And so up to 25% of the marine oxygen budget could be impacted by this net oxygen sink. And so this implicates that a lot of oxygen could be lost by going through this reactive oxygen species shuttle. So it could be shuttling a lot of oxygen around. So it's kind of an interesting thing to think about. So I'm going to just bring it back to, kind of coming to think about, how these reactive oxygen species are formed, why they're being formed, and then what are all the downstream and upstream reactions of those. And I'll end by saying that, I kind of like to end my talks with the idea that, opening our mind to thinking about reactive oxygen species and not just toxins. And there's Paracelsus, who is a Swiss physician in the late 1400s, who is the father of the field of toxicology, who said, all things are poison and nothing is without poison. Only the dose makes that a thing is not a poison. And hopefully today, I think I hopefully showed you that superoxide kind of falls under that umbrella of something that is not just detrimental for life, but also beneficial. And with that, hopefully I have time for questions, and thank you all for listening.
Thank you so much, Colleen. Virtual applause. We usually start the question period with students and postdocs. So if there is a student or a postdoc who has a question, then maybe you could turn on your video and ask it, or you could write it in the chat and I can ask it for you. Why?
Hello. Thanks, Colleen. That was very exciting. It was super exciting. I have a question. So you said that the superoxide production decreases as cell density increases, right?
Yes.
Do you have a sense of whether cells are actually detecting superoxide in their environment, or how are they sensing the cell density in their environment?
Yeah, that's a great question. I don't know. I don't know how they're sensing it. So that's a great question. So I think if in the fungal literature, it's been suggested that they're actually detecting hydrogen peroxide. So there's been a lot of research on hydrogen peroxide as a cell signaling molecule and sensing molecule. And so superoxide dismutates really quickly to hydrogen peroxide. So it could be that the same thing is happening there, that they're actually sensing the hydrogen peroxide. But yeah, we're just at the beginning. I don't know, but great question.
Cool, thank you.
Mm-hmm.
There's a question in the chat. Sarah, would you like to say it, or I can say it for you? Okay, I'll say it then. Or just chime in if I'm wrong. So it says, I attended a talk by Dr. Terry Hughes a few years ago, who suggested that Porites may be less able to grow in the face of bleaching events due to climate change than Acropora species in the Great Barrier Reef. So do you think this has any link to the higher superoxide concentrations you observed in Porites in comparison with the Acropora?
Oh, that's interesting. So Porites may be less able to grow in bleaching? Is that what I heard?
Yeah, I think that, yeah.
Oh, that's interesting, because Porites typically is considered to be such a resilient and hardy species. So our thinking was that these high concentrations of superoxide outside of Porites, I mean, they're huge, right? We had one measurement that was up to 300 nanomolar, which blew us away, but was that this may actually be like a protective mechanism for Porites. So like right now with the Skittle D outbreak in the Florida Keys and now in the US Virgin Islands of this stony coral tissue loss disease. We're thinking the Porites is one of the only species that isn't being infected, right? So we're thinking that maybe it's some sort of protective, barrier. And so we had done some work in Hawaii where all the Porites species there had huge concentrations of superoxide. And this was during 2014 when Hawaii had that massive bleaching event, and Porites didn't bleach, and Montipora bleached like crazy. And we had no concentrations of superoxide at the surface of Montipora. So we were actually thinking of it the opposite way. But we honestly don't know. So one of the things that we were really interested in doing is trying to figure out, what is the superoxide doing? We can speculate all we want, but we really don't have any proof right now. So we would love to take a species like Porites that has these really high concentrations, and a species like Montastria or Montipora that have no superoxide outside the cell. And then induce them to stress events, like whether that's a pathogen or a bleaching, and then see how superoxide concentrations change. You know, we can add superoxide, we can remove it, we could add a pathogen, we could, you know, mess with them a little bit and then see, you know, whether or not the superoxide is beneficial or not. I don't know if that answered the question. I don't think I answered the question at all. But
I think we can open up the questions to everybody. And Eldon has a question. I don't know if Eldon, you want to read it, or if you want me to read it, or if you want to just turn your mic on and your video on and ask it yourself.
Hi there. Hi, can you hear me?
Yep.
Thanks for the great talk. I just was thinking, it seems like superoxide is sort of a perfect signaling molecule, and I was wondering how it compares to other signaling molecules.
yeah, I think it's a perfect signaling molecule. It's reactive, it's short-lived, it doesn't diffuse very far. So it's good for, you know, signaling on short time frames, right? So it would be great for the outer membrane, right? So to be able to signal on the outer membrane. Hydrogen peroxide is the same. So that's some of the arguments that have been made for why superoxide might be acting as a signaling molecule on the outer membrane. Some of the others that have been suggested are nitric oxide. So NO has been suggested as a signaling molecule. How does it compare? It's also a nitrogen intermediate, right? And reactive. So I guess that's pretty similar. I don't know how it compares to other ones. Do you have other recommendations, other ones to think of?
I don't know, I guess totally out of the box would be something like ethylene. But I'm just thinking of the balance between toxicity and reactivity. I guess.
Well, NO is the same too, right? NO gets too high, it's toxic. And yeah.
Thank you.
So I think, Boris, go ahead.
Yeah, thanks, Colleen. This is, for me, particularly interesting because I know absolutely zero about the topic. So my question may be pretty mute, but I was interested in that you couldn't really pinpoint what the role of this extracellular oxygen species was, but you assume it has a role. Could it not just be some kind of waste product, or am I thinking the wrong way?
Yeah, so I didn't show the work that we've done with Thalassiocyra. We do believe it's a waste product. So it does show the typical behavior of it's a waste product, that by removing it helped it grow faster. And so that showed the typical behavior. With Rosiobacter, with Rosiobacter, it didn't. And I think a lot of that was because of the dilution behavior. We can't explain that it would upregulate superoxide production when you dilute it out because of a waste product, right? There was no reason. So Thalassiocyra doesn't behave that way. So Thalassiocyra actually behaved the opposite in that sense. There was another thing that Thalassiocyra did differently, and I'm blanking out on it right now. So, and also with the results that were found with E. coli, I think was really showing us that there was like these parallels with the bacteria, right? That with the bacteria, that they showed that you upregulate, you got these high cell densities, upregulated the enzyme that made extracellular superoxide, you got high cell densities. If you knocked it out, you could shut down growth, right? So I think there's something different going on between the phytoplankton and the bacteria for sure. So in the diatoms, we believe it's related to photosynthetic health. We believe that it's kind of like you're building up electrons and you need to dump them off, right? You dump off electrons, and that, you know, you remove that waste product, and that helps them grow. But with the bacteria, we can't explain that with the upregulation of both the decay rates and also the production rates within the dilution